Hello everyone, welcome to my channel Talks. This is Dr. Priyanka Bishnoi. In the previous lecture, we have covered macroscopic features of gingiva, and in this lecture, we would be covering microscopic features of gingiva. So let's get started. Gingiva comprises of the gingival epithelium and the connective tissue. The gingival epithelium is more cellular in nature, whereas the gingival connective tissue, it is more fibrous in nature. That is, it has more amount of gingival fibers and the ground substance. In this lecture, we are going to discuss this gingival epithelium in detail, and the gingival connective tissue we are going to cover in our next lecture. So let's get started with gingival epithelium. So as I have already told you that gingival epithelium, it is cellular in nature. So it has two types of cells, that is keratinocytes and non-keratinocytes or clear cells. The keratinocytes, they are present in large quantity, whereas non-keratinocytes or clear cells, they are further divided as Langerhans cell, melanocytes and Merkel cells. These three cells we will discuss in detail. Now coming to the function of gingival epithelium. What is the function of gingival epithelium? To protect the deeper structure. That is, it forms a microbial barrier. It will prevent the deeper structures by selective interchange only. And it achieves this means by the means of proliferation and differentiation. So proliferation, as the name suggests, it is the proliferation of cells. That is, there will be increased mitotic activity. There will be increased mitotic activity. That is, the cells will be dividing at a rapid rate. And this increased mitotic activity, this is seen in the case of stratum basal. This is seen in the case of stratum basal. As you already know, there are 4 layers of gingival epithelium. These are stratum basal, stratum spinosum, stratum granulosum and stratum corneum. So the maximum amount of mitotic activity, it is seen in the case of stratum basal. And as we go up in the layers, the mitotic activity will decrease. Mitotic activity will decrease. So here in this layer of stratum basal, you will see there is proliferation of cells. That is your keratinocytes. So these keratinocytes, they will be proliferating at a rapid rate. And the keratin produced here, the keratin that is produced here, that is of low molecular weight. That is of low molecular weight. So the keratin production is less and the mitotic activity is more in case of your stratum basal. And as we go up in the layers, the keratin production will increase. Keratin production will increase. Okay, now coming to the process of differentiation. So differentiation is basically a process of keratinization. The process of keratinization. So here you will see there are some morphologic and biochemical changes. So what are the changes that you will see here? That is the flattening of cells. Flattening of cells. That is, the cells become more flattened. So we see here in the stratum basal, the cells, they are tall columnar in nature. And as we go up, that is, as we reach the stratum corneum, the cell become more flattened. The cell become more flattened. So these are the morphologic changes that you will see during the process of differentiation. And as we go up, there will be no nuclei present. There will be no nuclei present in the upper layers as we move up. So these are the various changes that you see as the cells differentiate. Now, depending upon the degree of keratinization. Now, if you see here, depending upon the degree of keratinization, the epithelium can be orthokeratinized, parakeratinized and non-keratinized. So if the process of keratinization, it is complete, if it is complete, then it is referred to as orthokeratinized. If it is partial, then it is referred to as parakeratinized. And the third type is your non-keratinized. Now, how can you differentiate between an orthokeratinized epithelium and parakeratinized epithelium? As you see here, this diagram, here, this is your connective tissue and this is your epithelium. Okay, so here I have marked the 4 layers, that is your stratum basale, stratum spinosum, stratum granulosum and stratum corneum. So if you see in these 2 layers, that is your stratum granulosum and stratum corneum, the difference will be seen here. In case of orthokeratinized epithelium, the stratum granulosum is well defined, whereas in case of parakeratinized epithelium, it is not well defined. In case of stratum corneum, there are no nuclei present in case of orthokeratinized epithelium, whereas in case of parakeratinized epithelium, you will see pycnotic nuclei. There will be presence of pycnotic nuclei. So based on that, you can differentiate between orthokeratinized and parakeratinized epithelium. Now we are going to discuss all these 4 layers in detail. So this become more clear with the help of this diagram that you see here. Here I have marked the 4 layers, that is your stratum basale, stratum spinosum, stratum granulosum and stratum corneum. Now starting with the stratum basale. So if you see here, these have tall columnar cells. Stratum basale, it has tall columnar cells, and as I have already discussed that it has increased mitotic activity. It has increased mitotic activity and the keratin production is less here. Keratin production, the keratin produced here is in lesser quantity. Okay, and the keratin produced will be of K19 type, that is the low molecular weight keratin will be produced here. Okay, so now if you see, this is stratum basale, this is attached to the basement membrane or the basal lamina with the help of hemidesmosomes. Okay, if you see here, these are your hemidesmosomes. The one I have marked with the red color, these are the hemidesmosomes. So these hemidesmosomes, they will connect this stratum basale to the basal lamina or the basement membrane. This basal lamina, it is a 300 to 400 angstrom thick layer. Okay, and it is further divided into lamina lucida and lamina densa. The one which is close to the epithelium, it is the lamina lucida, and the one which is close to the connective tissue, it is your lamina densa. Now this lamina lucida, it has laminin type of protein, whereas lamina densa, it has type 4 collagen fibers. Type 4 collagen fibers. So basically the stratum basale, it has tall columnar cells. As you can see here, this is a tall columnar cell. Here the mitotic activity is more, that is the keratinocytes will be proliferating at a much rapid rate. Okay, and the keratin production is less here. The keratin that is produced is of low molecular weight. It is of low molecular weight keratin that is produced here in stratum basale. Now this stratum basale, this is attached to the basal lamina with the help of hemidesmosomes. This basal lamina, it is a 300 to 400 angstrom thick layer which is further divided into lamina lucida and lamina densa. The lamina lucida, it is close to the epithelium and contains a protein called laminin protein, and lamina densa, it is close to the connective tissue and it contains type 4 collagen fibers. Now coming to the stratum spinosum. Okay, coming to the stratum spinosum. Here you will see these are the large cells. These are the large cells. Okay, these are the large cells with small cytoplasmic process. So as we fix them, these cells will shrink, giving them their characteristic spinous appearance. Now this upper layer of your stratum spinosum, they have odd-land bodies. What are these odd-land bodies? These are your modified lysosomes, modified lysosomes, or you can call them as keratinosomes. These are your odd-line bodies. They contain increased amount of acid phosphate, which causes the destruction of membrane organelles. Okay, so this was about stratum spinosum and odd-line bodies. Now the third layer that we are going to discuss is stratum granulosum. So as the name suggests, there will be increased amount of keratohyalin granules that you will see here. See, if you see here in this diagram, the blue dots that you are seeing here, these are the keratinohyalin granules which are produced here. What is the function of these keratinohyalin granules? To produce keratin. They produce keratin. So here in this layer you will see three types of protein. That is your profilaggrin protein and locresin and laminin type of protein. Now what is the function of this profilaggrin protein? This profilaggrin protein, it binds the keratin fibers. And this locresin and laminin protein, they will form a cell envelope. This cell envelope, it will reinforce the outer layer and it will prevent water loss. Now coming to the fourth layer, that is your stratum. There is one more thing about stratum granulosum. We have already discussed about the proteins that you will see here. That is your profilaggrin protein, locresin and laminin protein. So the profilaggrin protein, it will bind the keratin fibers, and the laminin and locresin protein, they will form the cell envelope which prevents the water loss. Now one more thing that you will see here is the presence of your lamellar bodies. Now what does these lamellar bodies do? These lamellar bodies, they will secrete lipid in the extracellular space. Okay, so if you see these are the two cells. So the space here, this lipid will be secreted here in the extracellular space. So this also prevents your water loss. Okay, now coming to the fourth layer, that is your stratum corneum. This is stratum corneum. It has flattened dead cells. Okay, these flattened dead cells, what are these known as? These are known as corneocytes. Cells. So they are embedded in a lipid matrix of ceramides and cholesterol. So this is embedded in the lipid matrix of ceramide and cholesterol. So this was all about the four layers. So what are the changes that you have seen as we move up in the layer? As I already told you, as we are moving up with the layers, you can see the cell have become more flattened here. The cells in this stratum corneum, that is corneocytes, these are the dead flat cells. And the stratum basal have tall columnar cells. So this have become more flattened. Okay, the amount of acid phosphate activity, it will increase as we move up in the layers. And the third thing is the increased amount of tonofilaments. So these fibers that you are seeing here, these are the tonofilaments. They are made up of keratin, and the function of this is to provide strength. They also provide strength, support to the gingiva against the various mechanical stresses. So they also provide support. So this was the change. These were the changes that you see as we move up in the layers. That is, the cell become more flattened. The amount of acid phosphate activity will increase, and the tonofilaments will also increase as we move up in the layers. And the amount of keratin production will increase. Also increase, and the mitotic activity will decrease. There will be decreased mitotic activity as we move up in the layers. Okay. So now coming to the non-keratinocytes or clear cells. Now these non-keratinocyte cells or clear cells, as I have already told you, these are of three types. That is your Langerhans cell, then melanocytes, and third is the Merkel cells. Now coming to the Langerhans cell. These Langerhans cells, these are the antigen presenting cells. These are the antigen presenting cells to the lymphocytes, and they contain specific granule, that is your Birbuck granule or G specific granules. Okay, so this is the characteristic feature of your Langerhans cell. They are basically the antigen presenting cells. Okay, they are basically the antigen presenting cells. They contain G specific granule or Birbuck granule, and they are found in outer epithelium, and they are found in small amount in your sulcular epithelium, and in junctional epithelium they are completely absent. So this was about Langerhans cell. Now coming to the melanocytes, these melanocytes, they are present in the spinous and basal layer. Okay, and what is their function? They produce melanin. And the third one is the Merkel cells. So these Merkel cells, they are deeper in the epithelium. They are found deeper in the epithelium. They harbor nerve endings. And what is their functions? That is the tactile perception. So this was all about the gingival epithelium. Now coming to the 3 zones of epithelium. These 3 zones we have already discussed in the previous lecture. So let's revise it quickly. The 3 zones of epithelium, these were your outer epithelium, the sulcular epithelium, and the junctional epithelium. Now this outer epithelium, it is made up of free gingiva plus attached gingiva plus free gingival groove. So this outer epithelium, as you see here, this is the outer epithelium. This is made up of your marginal gingiva, attached gingiva, and the free gingival groove. This epithelium is keratinized. This particular epithelium is keratinized. It has retipegs present. As you can see, there are many retipegs which are present here in the outer epithelium. Okay, so this outer epithelium, here you can see it has all the 4 layers of epithelium that we have already discussed above, and the thickness of this epithelium is 0.2 to 0.3 mm. So this was all about your outer epithelium or your oral epithelium, outer or oral epithelium. Now coming to the sulcular epithelium, as the name suggests, it lines the gingival sulcus. Okay, this epithelium is non-keratinized. There are no retipegs present, and it is semi-permeable. That is, it allows the dead bacteria. So if you see here, the dead bacteria will seep into the gingiva through this sulcular epithelium, and the gingival fluid or the circular fluid that will seep into the gingival sulcus with the help of sulcular epithelium. So this is basically a semi-permeable membrane. Now coming to the third one, that is your junctional epithelium. That is the one marked with this green color is the junctional epithelium. So this epithelium is also non-keratinized. This is also non-keratinized. It is 3 to 4 layers thick, and as our age increases, it also becomes 10 to 20 layers thick. So it has 2 parts, the one close to the tooth surface and the other away from the tooth surface. So based on that, it is classified as internal basal lamina and your outer basal lamina. Now the length of junctional epithelium, this is important, that is the length of junctional epithelium. It is 0.25 to 1.35 mm. This is important, the length of junctional epithelium. Now how this junctional epithelium is formed? It is formed by the confluence of it is formed by the let me write it here for you. It is formed by the confluence of your outer epithelium or your oral epithelium with that of your reduced enamel epithelium resulting into the formation of your junctional epithelium. So while our tooth is erupting in the oral cavity, so as the tooth is erupting in the oral cavity, then there is confluence of oral epithelium with the reduced enamel epithelium resulting into the formation of junctional epithelium. So this was all about your gingival epithelium. In the next lecture, we are going to cover the connective tissue of gingiva, that is the gingival fibers, which is also very important. Thanks for watching my video. If you have any queries regarding this topic, then you can feel free to drop your comments in the comment section. Thank you.